we will start today by expanding our understanding of the scientific method, right? And so if you know what this is, and some of you might already, what type of structure this is, do not say anything, right? So let's work on this as if nobody knew what this was, okay? So if we are working on this and you take a look at this, remember, kind of some of the first things you want to do is make observations. So if you have observed this structure, right, here it is again. So just take a look at it real quick, and maybe you printed it out and so you can see it. But if you take a look at this, right, so your assignment, if you did it, if not, it's okay, was to derive 5 questions you would ask to determine what kind of organism is depicted in the image. So is anybody willing to share what those questions might be? You can just shout them out. Go ahead, somebody.
Is the
What are some of those questions you might have asked? Does it have a nucleus? Okay, I like that. Right? That's establishing whether it's prokaryotic or eukaryotic. Like that. What other questions might you ask? Are there one? Oh, I like that a lot, Brandon. Are there more than one organism pictured there? I like that a lot. What else?
If there's chitin in the cells.
Oh, there's chitin in the cells. I like that. So that's beautiful. That's a beautiful understanding of the carbohydrate, the polymer, chitin. Love it. Right. What environment is it found in? Like that a lot. Okay. Are there Are the cells bounded rigid? Okay, so that's getting kind of what Elijah asked also. Is, you know, is there chitin in the structures? Is the organism engaged photosynthesis? Love it, Brandon. That's excellent. Okay, very good, very good. Oh, what domain is it in? So, okay, so you're getting a little bit further than I wanted you at this point, Kelly, but thank you for thinking ahead of time, right? Do the cells have chloroplasts like it? So that goes back to um, you know, does it have a cell wall? There's a lot of good things there. Okay, very good. Nice job. Nice job. Okay. So are your questions? So some of you might have been using inductive or deductive reasoning. So if you used inductive reasoning, what conclusion have you drawn from your ideas? What kind of conclusion have you drawn? Anybody want to mention anything? How the organism grows on a tree? I like that conclusion. That's very nice, right? So very nice. So let's do deductive then, right? And so, um, what might be your general premise of what you're looking at right now? What might be your general premise? What's your understanding? What's your understanding? Or maybe we use the same one from earlier, right? It's growing on a branch. Okay. So from those kinds of ideas, could you provide a hypothesis for what type of organism that you are observing? Could you provide a hypothesis? What might your hypothesis be?
That it uses the tree to provide energy for itself.
Okay, very good. So it uses the tree to provide energy for itself. All right, good. So whether or not that is true or not, we have to test for it. So how might you go about testing that hypothesis, Elijah?
Um, Using a process to like figure out how it makes ATP, whether that is through chlorophyll or whether it's through just like eating the tree.
Perfect. Perfect. So in that So that's a really good one. So if you think about that for a minute, right? Um, and let's just say that we moved ahead in your hypothesis. We tested to figure out whether or not um, the organism produced energy through photosynthetic means or not. So these are the things that we found out, right? And I'm just kind of jumping ahead a little bit. So these are the kind of things we found out. Number one, the organism is partially photosynthetic. But the other thing we found out was that it's attached to the trees, but it's not really parasitic on the tree. So now, what did we find out? What does that lead us to understand? Oh, it's symbiotic. Okay, so can you, Alexandra, can you expound on what you mean? Are you talking about there's a symbiosis with the tree, or are you talking about some other symbiotic relationship between the two? So between two, so you're already jumping to the conclusion that there are two organisms associated with the organism, right? But let's go with the other route. Let's say there's a symbiotic relationship with the tree. So if I tell you then, does anybody know the three types of symbiotic relationships? Can anybody tell me what the three types of symbiotic relationships are?
Commensalism,
Commensalism, like it. Can you tell me what that means, Aiden?
one benefits, but the other doesn't, but it doesn't cause any harm to the one that does not
Good, I
benefit.
like that. And so that's commensalism is one. Can anybody name the other two types of symbiotic relationships? Mutualism, okay. Annabelle, do you want to give us the other one is parasitism. Okay, so maybe to save time, maybe I just kind of give the definitions of them. Mutualism is the relationship between two organisms, and both of them mutually benefit from the relationship. Parasitism is a relationship between two organisms. That's all right. And one of the organism benefits from the relationship, but um, the other one is harmed by the relationship. Parasitism, right? So now we go back to the original hypothesis. You know, looking to see we were saying that the organism is taking nutrients from the tree. Well, our observation now has been that the organism is commensalistic, right? So it's not taking anything from the tree itself. It's just simply growing there. It's commensalistic, right? So that goes to um, maybe a little bit more information. And might you adjust your hypothesis? What would be the adjustment to the hypothesis? What might be the adjustment to the hypothesis? Anybody?
I would probably look at the chitin next.
Okay, so you want to look at chitin. Okay, so we proved that there's chitin in the organism, right? And so if we say that there's chitin in the organism, what groups of organisms have chitin in their cells, Mr.
It's like
Elijah?
fungus, right?
Fungi, yeah,
Fungi.
and sometimes algae do also, right? Good, like it. But we know that there's definitely chitin in the cell wall, right? So now, what might be your what might be one of the conclusions you might reach? What might be one of the conclusions that you might reach?
That it belongs to that. Um, I forget the name.
So it belongs to at least somewhat part of the fungal kingdom, right?
Yeah, the
And
kingdom.
really, does anybody know what the organism is? Does anybody know what that organism is? Or it's really three organisms. So It's a lichen. Let's forget. It's a lichen. A lichen is a combination of an alga and two fungi, right? And so what happens is, if you think about them, they're all different kinds of organisms out there that are lichens. And a lot of people go through their entire life and not really even notice these things, right? One of the really cool things about this is the algae themselves photosynthesize, right? Because the fungi are not photosynthetic, but they produce carbohydrates by sequestering CO2 out of the atmosphere, reducing them to form sugars. And then the algae and the two fungal species that are associated with this symbiotic relationship, they're in a mutualistic relationship, um, will all benefit from them. At the same time, the fungus is providing protection from the algae so that the algae are not harmed, right? And that's pretty cool. These are epiphytic, right? These are growing on the surfaces of this tree branch, but they're not hurting the tree branch. And they can be found in lots of different formats. These are just things that I took from my backyard. I just took these pictures from my backyard. They're growing all over the place. A lot of people, including me, will tell you that the lichens will only grow in places where the the atmosphere is not polluted because they need oxygen, right? And if there's, you're not going to find these in downtown San Antonio, right? San Antonio is a pretty polluted town. You're not going to find these in downtown San Antonio, but the further away you get from San Antonio, the more likely you're going to find them. But they, in my backyard, they're all over the place. They're even growing on my fence, right? So you can see them growing on my fence, right? Here's a close-up view of them on my fence. They're epiphytic, so they don't need to be attached to anything that is organic or anything that's animate, right? They just simply can attach to any structure because they're just using those structures as a means to lay down their foundation, right? The the three organisms that are part of this mutualistic relationship are taking care of themselves. Okay, now they are a food source for birds and for other organisms, but for the most part, they just add beauty to nature, and they help to sequester um, they help to sequester CO2 out of the atmosphere. So they're doing a good job of helping to cleanse the environment, right? And so you can see how I just took a whole bunch of pictures all over my yard. Hey Elijah, what's up?
So if lichen grows on inanimate objects, does it break those down and turn them into like dirt?
Nope, nope. They are not absorptive at all. I mean, I guess they could be, Elijah, because there are two fungi that are in the relationship. But as we understand it now, those fungi are mainly there to protect the algae. Yeah, it's a pretty cool relationship. Okay. Anybody have any questions? All right. So that kind of helps you, I hope, to understand the kinds of questions that we should be asking as scientists, right? And so you should be doing this every time we're together, right? You should be asking questions to yourself about what does that mean? You know, Science is about really discovery, right? And so a lot of things that are scientific theory are dogmatic. And that just simply means that that's our best understanding of what that science means today. That doesn't mean it's not going to change in the future because our as we get more information, we can change the way we look at different ideas. And that's really one of the coolest thing about science is that we're forever learning and therefore we're forever changing our ideas about how biological mechanisms work. Some of the mechanisms that we know, some of the theories that are out there, right, haven't changed for a long time. But that's simply because all of the research that has been done just simply continues to support those initial ideas of of how those mechanisms work, right? So remember, we talked about the theory in science and the theory when people use it in everyday language, right? When people use theory in everyday language, it's just simply a guess, right? But when we use theory in science, we say it's a scientific theory. The theory of evolution, right, means that those ideas, those tenets of evolution, continue to be proven by numerous studies that are out there, right? A mountain of data that basically says this is the way it is. Okay. So the last time we were together, I told you that I would probably end up breaking up the PowerPoint that I had, right, to separate out the phylogeny and the taxonomy and the classification, so that we could address that pretty quickly. And so that's what I did for you all. So if we think about this for a minute, right, phylogeny, taxonomy, and classification of organisms, right? So um, taxonomy is how we put things in order and name them. Classification is how the techniques that we use to put these things in these different groups. And phylogeny is how everything on the planet is related to everything else, right? Because we as humans and everything that's on this planet shares DNA, right, with everything, right? And that just means we have a common ancestor. And so this is not a discussion about evolution. We will have a discussion about evolution at the end of the semester, but really this is just looking at how things are related to each other, right? And so it all starts with the molecules, and we're going to be talking about molecules after this particular talk on phylogeny, classification, and taxonomy, right? But if we think about molecules, it's just simply um, at least two atoms coming together to form a molecule. And some of the molecules that we're going to be talking about are going to be very, very complicated, right? And so you can see that there's some different molecules here. And so a lot of these are stick figures, but others you can see are space filling models, right? You guys have seen these before. And then if we think about just one of the most complicated um, molecules that there is of course DNA. And so you can see how it's arranged in a double helix, right? And it has, as almost all biologically active structures, molecules, they are mostly consisting of HONC, right? HONC, which is hydrogen, oxygen, nitrogen, and carbon. And when you think about that, right, those four elements themselves, those four elements make up about 96% of everything that's biological, right? So including you, right? Those four elements themselves, right? If we continue to think about life, then really um, all cells are derived from other cells, as we know, right? And they when they are dividing, either through mitosis, through meiosis, or through binary fission, they inherit DNA from their parent cells, right? Or their parent organisms. And of course, this DNA has got chromosomes in it, and they've got genes in them, and the genes themselves really code for specific proteins. And so really, when you think about it, DNA itself is the master molecule. It directs essentially everything that happens within the cell itself, okay? And so again, if we think about this, right, a lot of times when we think about replication, people are always thinking about, you know, parents coming together to form a child, right? But most of the organisms that are alive on the planet do not undergo any kind of sexual reproduction. They just sexual reproduction, where we as humans, we have both sexual reproduction, right? We form gametes, either sperm or ova, and then we also undergo mitosis, asexual reproduction, in that all the cells, all of our other cells that are part of the body, the somatic cells, right? Whether it's your skin cell, your liver cell, or whatever, those undergo mitosis, right? So they're just simply making clones of each other, but DNA in them. So the DNA in them will actually is the information that if we collected something. So if I was really liking I don't know, an individual, let's say Brad Pitt, and I really like Brad Pitt a lot, and I wanted to have my own Brad Pitt, I could walk around and follow Brad Pitt, stalk him, if you will. And let's say he drank a cup of coffee, but he threw his coffee mug or coffee cup in the trash can. I could go in there, collect that coffee mug, and take some of his epithelial cells that he left behind. And we have the technology now to clone that individual, right? So we can do that. Some countries have done it, right? It's not supposed to happen, but some countries have done it, right? And so it's an amazing thing how advanced our um, our understanding of DNA has become, that we can apply all this information and do all these really great things, and in some cases maybe not so great, depending on what you think, right? But if you consider just that molecule of DNA, right? Um, it's double helix, right? There are several nitrogenous bases that are part of that particular double helix, and those, of course, are adenine, thymine, guanine, cytosine, and so they pair up, and so then you have the DNA, and then you have really the molecule that directs everything in your body, right? So that DNA can be used for lots of different things. I don't know if you know this or not, but nowadays you cannot hide from science, right? If we have DNA at the scene of a crime, we can use that DNA to figure out who committed the crime, or at least who was in the area when the crime was committed. But the thing about, in order for us to find out, is your data, your DNA has to be within one of the databases that we have access to, right? So if you've done 23andMe or something like that, I have, your DNA is now on file, and really, the FBI or whoever could ask for, with a warrant, ask that for information, and it has been done in the past, where we could use that information, that huge sequence, right? That huge database, to figure out who might have committed the crime. But we don't even have to do that, right? We can just look at, if we think that you are at fault, we can go and we can either collect your DNA by you just throwing it away. If you throw your DNA away, say on a coffee cup, it's no longer your private property. You have disposed of it. It belongs part of the public domain. So they can just follow you around and take that cup. Or they can um, get some information from one of your sisters or brothers or cousins or whatever, and they can use that information to help to show that you were the culprit of the crime, right? It's pretty cool the way that works, right? But we can also use that DNA to do all kinds of other things, including studies of biodiversity, right? And so, yeah, we're going to be talking about DNA a lot in this course, right? But we're going to also prepare you. I'm going to prepare you to go on and talk about the larger organisms, right? This course, 1406, is all about the cellular and the molecular mechanisms, but 1407 is structure and functional organisms. And so you're going to go over there if you're going to be on a science path and not an applied health path. So you're going to go there and you're going to take 1407. You're going to learn a lot about the morphology and the structure of different organisms, right? And so this course will help you get there. We have to understand really how these different organisms are classified, how they're put into groups, right? The taxonomy. And so one of the ways to think about this is to think about how organisms were first put into groups. And so it all started with Carolus Linnaeus. And so Linnaeus, a long time ago, was more of an observational scientist. He didn't have the tools necessary to be able to look at the ultrastructure, the molecules, not even the cell, right? So he was mostly doing observations. And what he noticed was that everything was either associated with plants or animals. At least that was his initial thought, right? Many people have come after him. Anton van Leeuwenhoek, um, Robert Whitaker, Carl Wise, and Fox. They've all come afterwards, and they've added a lot to the ideas of what classification is, right? But if we can think about, this is one of the earliest, after Linnaeus thought, well, everything's plant or an animal. Well, then people came along and they said, Whitaker came along and said, well, there's lots of different groups of organisms, right? And so he said things are either plants, they're animals, they're fungi, or there's these single-celled organisms that are eukaryotic, that are protist, right? And then he said, but there's also the bacteria that are more common, right? The E. coli and the Staphylococcus aureus and the Streptococcus pyogenes and the Mycobacterium tuberculosis, all these organisms that we hear about because they cause infection or they do something to interact with the individuals, right? Those are bacteria. But he said there's this other group of bacteria that are called the archaeobacteria. We just simply call them archaea, that are bacteria also, but we never talk about them so much because they're in the environment. A lot of them are, even though some of them are found in your backyard, a lot of them are found in these really extreme conditions, right? And so therefore we don't really talk about them very much unless we're researching them. But they're bacteria. So really, when you think about that for a moment, right? These were scientists who were postulating that the things on the top of this blue line were eukaryotic, and the things that were below this blue line were prokaryotic, right? Somebody remind me what the difference between eukaryotic and prokaryotic are, real quickly. Somebody just say something.
Nucleus.
Nucleus, okay, I like that. So which one, eukaryotic, prokaryotic, has a nucleus?
Eukaryotic.
Eukaryotic. Very good, Colin. Very good. So the prokaryotes are going to be on the bottom, and the eukaryotes are going to be on the top. So where's the emphasis on those initial ideas? Where's the emphasis? Eukaryotes or prokaryotes? Where's the emphasis?
Eukaryotes.
Eukaryote, for sure, for sure, right? We were more interested in them, and really, from a lot of aspect, from perspectives of many people, the eukaryotes are still the things that were of interest, right? Because we like cats and dogs and snakes and birds and all these different things. Those are all eukaryotic. Very few people, unless you're somebody like me, likes the bacteria, right? I love the bacteria. But that's not my favorite group. The fungi are my favorite group, and I'll explain why later, right? And so as we started to understand these things, we came up with ideas of how things are put into groups, right? And we came up with K for kingdom, P for phyla, C for class, O for order, F for family, G for genus, and S for species. And this was the first order of classification that became widely accepted. For a long time, this is the way it was, right? But it wasn't until we started to get a lot of understanding of the molecular techniques that we started to see the differences of these organisms. Yeah, they're pretty similar, right? But also things that you think are going to be the same uh, end up being different, right? And we have to be careful with that because we don't want to stress the differences in just one gene, but we also want to be able to stress the differences that make these organisms unique, right? And so it was until much later in the 1970s that Carl Linnaeus started talking about these super kingdoms. And he said there are only three super kingdoms. And he said that things were either bacteria, they were archaea, or they were eukaryotic, right? And so for a long time, I was teaching two different types of classification systems, right? And so we finally, as scientists, came to the conclusion that that Carl Wise and George Fox, they were really onto something. And so we needed to incorporate these super kingdoms, but we didn't want to call them super kingdoms. We wanted to give them another distinction. Does anybody know what that distinction is nowadays? What is the top of the hierarchy nowadays?
Domain.
Domains. Very good. Domains. It's the new kid on the block, right? Because as we learn, remember, science is about understanding and applying. And as we get more information into our scientific repository, then we make judgments and we might make changes to what we know, right? So before, you guys told me that eukaryotes were really the focus. They were really the kind of the things that we gave a lot of credence to, right? And so if we move along, then we find out that you can see that here's a taxonomy and including the domains, and you can see that as we go from an area of most inclusivity, the domains, and go kingdom, phylum, class, order, family, genus, and species, that things become very, very specialized. They become very, very unique. Okay. Now, I'm not going to expect you to know the phyla, class, order, family, but I will expect you to know, because you're scientists, what a genus and a species are. I'll expect you to know what the domains are. There are 3 of them. We'll talk about them in a minute. And then I'll expect you to know the kingdoms, at least uh, what's most accepted right now. There are many, many ideas about kingdoms right now. But what is most accepted, of course, is that within the eukarya, there are 4 kingdoms. Okay? So if we look at one of the earliest ideas of phylogeny, of the phylogenetic tree, how everything is related to each other, you can see that you have the bacteria, the archaea, and the eukarya. But looking at this particular first early idea of a phylogenetic tree, where do you think the emphasis is on? What's the emphasis? What do you think? Anybody? Anybody tell me what the emphasis is? What is the emphasis? So okay, Tammy, that's a good thing to say, but that is the truth. But if you're just looking at this and don't have any science background, you might basically say, well, they all kind of look the same, right? So this was our early idea of what a phylogenetic tree looked like, right? Without understanding how these things are used, right? How the clades and how anything's used, right? This is one of the first early ideas. More currently, about now 12 years ago, the most current phylogenetic tree was published, and here it is. So now, check it out. This is the domain to which the bacteria belong to. This is the domain that the archaea belong to. And this, ladies and gentlemen, is the domain by which the eukarya belongs to. Opanstacanta, right here. We are somewhere right here, okay, as humans. All right. So now, based on what we know, based on the current phylogenetic tree, and based on the numbers, the diversity of the organisms on the planet, where is the emphasis now? Tammy,
Bacteria.
the bacteria. Hmm. Yep. There are many, many more in numbers and variety of bacteria than anything else. Isn't that interesting? Look at the eukarya. They're the new kids on the block, right? Uh, they've been around not as long, okay? And when you think about this, right, what do we have a common ancestor with? Which other domain does the eukarya have a common ancestor with? What do you think?
The archaea.
Uh-huh. Yep, right here. So what that means is that we share common ancestors. We share common traits, an ancestor with the archaea. We're more closely related to the archaea than we are to the bacteria. Isn't that interesting? Right? And some of you may have heard um, of the theory of endosymbiosis, right? Where we believe that early cells uh, worked in symbiotic relationship, and eventually some of these cells inside of other cells became organelles, and then these cells that had these organelles became um, the more complicated eukaryotic organisms that we see today. Okay, phylogeny shows you how everything on the planet is related, because everything that's alive on this planet shares genes with everything else. We share genes with bacteria, right? We share genes with protozoans. We share genes with pigs, which is a very close relative to us, right? Even things that are not living can be traced phylogenetically. So name something that is non-living, because this is a phylogenetic tree of things that are alive. Name something that is non-living. Rocks. Okay. But they have to be organic, right? What dead organisms are dead. They're not alive. Dirt. Oh my. So okay, Connie, I get
Virus.
it. Virus is what I was looking for, right? Viruses are non-living, right? And so if we think about this, uh, this is the phylogeny of a very important virus. Do you know what this is the phylogeny of?
COVID.
That's right. This is the phylogeny of COVID. Look how many times it has mutated. We've only heard of three of them, right? SARS-CoV-2, then we heard of the Delta variant, and now we're hearing about the Omicron variant, right? Well, we hear about those, or you all hear about those, or the common folks hear about those, because those are the ones that are the most destructive to us. But a lot of us have been looking at the different mutations that have been occurring in the in SARS-CoV-2 and COVID-19, and it's vast, thousands and thousands of mutations, right? But most of them do not change the infectivity or the dangerousness. The term we use for dangerousness is virulence of the virus. The ones that have changed drastically, we talk about them, the Delta variant and the Omicron variant. Okay, but here is the whole phylogeny as we're looking at the different variants of COVID-19. And this is older, right? This is no, I don't even think it's here. I don't even think it's here, because this, again, look at this. This was published back in April 2020, and it's the only one I could find. That's the most current one I could find, Alex. But so I don't even know. I don't even know if it's on here. Okay. Yeah. All right. So if we talk a little bit more about this, remember the three domains are the bacteria, the archaea, and the eukarya. The viruses do not belong to a domain because they're not alive, right? And so that's really important to understand, right? So the three domains of life are just simply um, the archaea, the bacteria, and the eukarya. And then within the eukarya group, right, there are some kingdoms: plantae, fungi, and animalia, and protista. But now the protista is going under a drastic overhaul. Some people want to add as many as 15 different kingdoms that once made up just the protozoan, because there's such a vast group, right? So right now it might be a few years before we decide what we want to do. But so the four different kingdoms right now that are mostly accepted. Some people have moved on, right? Some scientists will say, okay, I'm over here already. I'm at 8 kingdoms, right? Other people are like, there's just not enough data to say that there's 8 kingdoms, but we know that there's 4. So that's what I'm going to talk about. I'm going to talk about the 4. And those are, and when you go to 1407, you're going to see how these things are broken up even more. And I'm going to let them talk about that. That's not really the scope of this course, right? I'm giving you a foundation of the cellular and molecular. So right now, I'm going to tell you that's what's most accepted is plants, animals, fungi, and the single-celled organisms that are eukaryotic, and we call those things the protist or the protista. Okay, good. So now, as we think about these things, right, I want you to be sure that you know what the scientific name is. The scientific name was first hypothesized by Carl Linnaeus. That Linnaeus guy was ahead of his time, right? Not only did he say, hey, there are 2 different groups of organisms, plants and animals, but he said everything should have a proper name, right? He called it the binomial nomenclature system, right? The 2-name system to name things, right? And he started out with the genus and the species, right? This is my late puppy, my dog. Her name was Bandy. She passed away um, from cancer. But you can see, I don't know if you can see it. Maybe you cannot. But she um, she and I were playing in the backyard. She ran and she tore her ACL. We had surgery to fix that, and then she developed necrotizing fasciitis, flesh-eating disease. And my wife and I took care of her um, for about 6 months. We we cleaned her wound. First of all, she was treated with antibiotics and got that treated, right? I had a hand on what we were going to do there because I knew a lot about antibiotics. So the vet and I, we problem solved. We got her so that she didn't have the infection. She almost died 3 times. But then my wife and I, because the vet wanted to charge $60 every time that they would do hydrotherapy, and I just simply said, no, I can do this at home. So my wife and I did that. I have videos of my wife and I doing hydrotherapy on her, right? But so we cleaned her wound twice a day. We dressed it twice a day. My wife is very, very creative. We couldn't find any dressings large enough to cover the wound. So we tried to use an Attend, right? But that made her too warm and she developed a yeast infection, a super infection on top of the bacterial infection. And so we treated her for that. And I just simply treated her by using Tinactin from Walmart, got rid of the yeast infection. The antibiotics took care of the bacterial infection. And then my wife came up with this kind of little covering that she sewed that we could attach a telfa on the inside of it, and that was became her covering for her wound until it healed, right? And so she survived, and she lived another 8 really great years with us before she died of cancer, right? But getting back to the nomenclature system, right? So Canis familiaris is the genus and the species of the domestic dog, right? Canis is the genus and familiaris is the species, right? For For humans, it's Homo sapiens. Right? Homo being the genus and sapiens being the species. The rules of the binomial nomenclature system is that you can abbreviate the genus by simply putting the first letter capitalized with a period after that, and then sapiens or the species name afterwards. So Homo sapiens is the same thing as H. sapiens, right? How many people have heard of E. coli? How many people have heard of E. coli?
I
Yeah.
have.
Okay. Yeah, it's the most commonly studied organism, right? Good. Hey, Elijah, did you want to say something, or you're just raising your hand that you heard about it? Oh, bluebell. No, that was Listeria monocytogenes. That was different. Okay. Salad, yeah. Salad, all kinds of different things. But most E. coli are not dangerous, right? Oh, okay. Don't even get me started with Chipotle. Okay. Don't get me started there. But yeah. So and most people think E. coli is bad, but only certain strains of E. coli are bad, right? Not most of them are not bad. But does anybody know what the E stands for in E. coli? Senor Connor, what say you? Connor's being shy today. That's right, Escherichia. And you even spelled it correctly. Look at you. That stands for Escherichia. Escherichia coli is the same thing as E. coli. Okay? Please know the difference so that if I show you a scientific name, you know the difference between a genus and a species. Okay? Anybody have any questions? All right. So, the unity, right? We've been talking about, oh, it's okay, Connor. We were talking about, how everything on this planet shares DNA, right? And so here's a great example. If we took the ciliated cells from a single cell parasite called Paramecium and compared it to the ciliated cells of our windpipe, our, if you will, our trachea, what we're going to find out is genetically they're similar. What does that mean? What does that mean? That the DNA from these two different structures, the cilia that are found surrounding the paramecium and allows it to move, and the cilia that's found in around our trachea. What does that mean? They're genetically similar. What does that mean? What does that mean? Anybody? What does that mean? That means we share DNA with a single-celled protozoan. That's what that means. Okay? And we can use phylogeny to study uh, things that have been on Earth, right? Um, we have we can look at fossils and we can carbon date them, but also in some of the organisms that have been nicely preserved, right? In amber or in the frozen tundra of the Antarctic or Greenland, as this frozen tundra starts to melt, some of these organisms are becoming um, available to us, and so we can study them. And I don't know if you know this, but there's a project right now going on in Siberia to bring back the mammoth, the woolly mammoth. You know, I'm just not for that at all. I think if an organism went extinct, there was a reason it went extinct, and I don't see a reason to bring back those organisms because, of course, the thing that's going to follow that is let's bring back a T-Rex, and that's just going to be not a good idea, right? The,
Would,
you know, go ahead,
I was just gonna say,
Aiden.
wouldn't you think that it depends on the organism that is being brought back?
What do
Because
you mean? Explain what you mean by that.
I mean, I see why not a woolly mammoth because its natural size and how much it ate,
Uh-huh,
it would take up a lot of space and it would
uh-huh.
change a lot of ecosystems if there was more than one of them. But like smaller species or ones that don't have a grand impact on the ecosystem around them, wouldn't you?
Everything you bring back, Aiden, is going to have an impact on the ecosystem,
Right,
right?
but to uh, not as a great extent, I suppose.
Well, I don't know. Are you going to bring back a compy? If you bring back a compy, those things are going to take over everything. Are you with me? And compies aren't very big,
What is a
right?
compy?
Oh, it's got a big old compo something, big old name, right? Look up compy on, and you'll see it.
Okay,
I don't know. Did you watch Jurassic Park? Did you watch Jurassic
uh,
Park?
long time ago.
Okay,
The
those
original.
little tiny lizard things that were chasing people and biting them, those were compies.
Oh, okay, okay.
Okay, so if you bring those back, whew, be a problem, right? If you bring a velociraptor back, not as big as a T-Rex or a mammoth, there's going to be some problems. What are you going to feed these things? Are you with me? So let's just not go there. How
Okay.
about do this? How about do this, Aiden? How about we concentrate on the species that are here and help them survive? How
That
about if we
is
do
a
that?
that is a good example, yeah.
Okay. So cheetahs are in trouble, tigers are in trouble, elephants are in trouble, the vaquitas in trouble. Why don't we concentrate on those guys and change the way we do things on the planet, and then use our scientific uh, methods to help them survive? Are you with me? Because the cheetah is now bottlenecked, right? There's only a few organisms on the planet, right? Um, and so, oh, you're going to be in conservation, Kelly? Good for you. So yeah, the cheetah is bottlenecked now. So there's only a few, and maybe not even, maybe I don't know. All the cheetahs might be related to each other in that there's just no variation in the DNA, right? And so I don't know. I think we ought to try to figure out how to save those organisms first, right? And then let's worry about the mammoth. I have no idea why they want to bring back the mammoth. It just doesn't make sense to me. And there's a whole article. I might send you that article that I read about bringing back the mammoth so that you can read it and maybe we could have a discussion about it later. But
Right.
it is just, it's
They've
just,
been on the mammoth for years though. It's been like since like
oh, I know, but do you know
the
why?
90s.
Why would they bring them back?
Yeah, I don't see
For
why.
food, maybe?
Yeah,
I don't
yeah.
know. Maybe they make a scrumptious steak. I don't know.
Everybody likes
Nobody
exotic.
knows that, right? Well, perhaps. Yeah. So we're going to be faced with a lot of challenges, right? As our climate gets warmer and the frozen tundra continues to melt. I'm afraid this is my biggest fear. I'm afraid that there's going to be some infectious agent that killed off a whole group of species of organisms that's going to be unleashed on humanity, to which if you think we weren't ready for COVID-19, one of these things could just devastate us completely, right? Think of something like Ebola, but it's airborne. Yeah, that's my worry, right? Yeah. So we can even look at infectious agent plasmodiums. Anybody know what plasmodium is? Sir Connor, what's plasmodium?
I honestly don't remember the definition, but I do know that I
Okay,
learned it at some point.
okay. It's malaria. So we now can study malaria and figure out how we might be able to change the organism so it doesn't kill millions of people on an annual basis. Now, we have to be careful when we start doing these things, right? Because if we start to interfere with the natural progression of life on Earth, right? I mean, there's already about 8 billion humans on this planet. And if we start to make changes in the DNA so that less people die, very quickly we're going to run out of resources, right? It already takes Mother Nature about 1.3 years to replenish the resources that we as humans take off this planet on an annual basis. It is not sustainable. So if we continue to do this and continue to help people survive, then we're going to be in trouble. And sometimes science and scientists have tunnel vision. Oh, this is good. We can cure cancer. Now, don't get me wrong. Curing cancer is a good thing. But if you cure cancer, then less people are going to die. If you cure malaria, then less people are going to die. And we're going to have a lot of people on the planet, all of which are going to be competing for the same resources, right? We're going to have to get better about taking care of each other and feeding each other than we are now. Because how many of you believe that we as humans are in competition with each other for resources? How many of you believe that? The answer is yes, right? If you go to HEB and buy a month's supply of food and put it in your refrigerator and freezer, you've got a little extra money because a lot of people, even in this country, can only buy things for maybe 2 or 3 days, right? So who's winning that battle, right? And there are places on this planet where people might earn $4 a month. So who's going to win? Who's going to win that? Who's going to win that challenge? The people who currently have, correct, the people who have all the resources, right? So we have to be careful that that we watch out for each other, right? And to me, science is important, but again, we need to not have tunnel vision and we need to be thinking about the larger picture of things before we go and do crazy things, right? Are you with me? So here's an example of that. About 5 years ago, when Zika first hit the United States, there were people who, using CRISPR technology, wanted to go and take the population of mosquitoes and make them sterile so they wouldn't replicate. And people thought, oh, what a great idea. We can get rid of Zika. Okay, so I told them at a conference I was at a nerd conference, I said, how many cases of Zika have we had this year? And somebody said 6. And said, okay, so you want to wipe out the mosquito population in South Florida to not allow for 6 people to not get Zika? Why don't we educate the people who contracted Zika to be able to resist coming in contact with the mosquito instead of getting rid of the mosquito? Now, I don't like mosquitoes all that much, but why am I against getting rid of mosquitoes? Why am I against getting rid of mosquitoes? That's right. It's a food source for many other organisms. And if you get rid of the mosquito, you're going to get rid of a lot of other organisms also. That's what I mean by being careful about wiping out one organism, because evolution teaches us that if you get rid of one organism from this planet, another organism will take its place. Are you with me? So you get rid of the mosquito, but let's say the simulate, the black fly, the black biting fly, Yeah, they are. They're important pollinators also. But let's say the black pollen, or I'm sorry, the black biting fly takes its place, and now you have these flies that bite, and when they bite, you know it, right? The mosquito, you might not even feel it, but the biting flies are a whole different group of organisms, and they can take up that niche. And what happens if they carry Zika, right? Because now Zika needs a new host, and Zika decides, maybe I'll mutate. Viruses don't think that way, but let's just say they mutate, and the Simulidae becomes the new vector, right? Now we just got rid of one organism and another one took its place. This is why we need to be careful. Okay? Any questions about any of the things we've talked about so far? So it's important to understand that it's really about not just understanding the science, but it's really about what happens if we do something? What are the ramifications of doing that, right? And so we just need to be careful, okay? So any questions about anything we've covered so far? I've got about 20 minutes or so. We're gonna start chemistry. We're not gonna finish it, I don't think. Maybe I will, but we're gonna start it. And this is gonna be review for a lot of people, right? But I want to be sure that we're all on the same page. One of the cool things about ACC is we got people in here that understand this, as this is gonna be boring to them, and we've got people right now in this classroom that maybe are thinking about uh, a career in science, who maybe have never seen this stuff before, right? And so there's lots of different ways to talk about atoms and elements and the bonding of them, right? I'm going to do it from a biological perspective, right? And so that's going to make it easier for a lot of people to understand. But if you take chemistry, they're going to do it from a chemistry perspective, and they're going to be talking about specific orbitals and shapes of orbitals and things like that. I'm not going to do that. I'm just going to simply show you how things are put together. Okay? Let's get started. So if we think about this, right, for this part, I want you to be able to look at the structure of atoms and molecules. I want you to be able to draw simple molecules. You should already be able to draw atoms and their orbitals and their outer shells. If you don't, we're going to do that together here. But I want you to be able to draw some molecules, right? And show me the interaction of the atoms and those molecules. I want you to be able to describe chemical bonds and their interactions. So when we think about this, we're mostly talking about covalent bonds, but we could also have ionic bonds. And also we could have hydrogen attractions. Some people will call those hydrogen bonds, which is fine, but they're really not bonds. They're more of attractions than anything. And we're going to talk about that. We're also going to be looking at acids, bases, and neutral pHs. We're going to be looking at the chemical reaction and the functional groups, and how the addition of those functional groups to a molecule changes the way those molecules interact. We're going to be looking at properties of water and why water is important and how water works the way it is, right? And then we're going to be looking at the biomolecules or the macromolecules and talking specifically about how they function within the cell. We're going to be looking at carbohydrates, lipids. We're going to be looking at nucleic acids, right, including things like ATP. And then we're going to look at proteins, right? So this is for the next 2 weeks, we're going to be doing chemistry or some aspect of them. Okay? Any questions? All right. So again, if we think about the building blocks of all molecules or atoms and the way that they interact with each other is the way they form bonds, right? Some of them like to donate electrons, some of them like to receive electrons, some of them like to share electrons, some of them like to share electrons equally, some of them like to share electrons so that they bogart those electrons, right? They take those electrons and they keep them for themselves, and every once in a while they share it. But these are the things we're going to be talking about, right? And if we look at the periodic table, I hope you all made a copy of the periodic table. We may not get to working too much with it today, but for sure we will on Wednesday, right? And so the periodic table is the table of elements as they are found in nature, right? If it's not on this particular table, if there's any kind of variation, those are called isotopes, right? And isotopes are really cool because isotopes all want to be what's naturally occurring in nature. And so in order to become that, they have to release energy. And when they release energy, we can harness that energy for good, sometimes for bad, right? We can make atomic bombs with those radioactive materials, but we can also use them to carbon date. We can use them for PET scans. We can use them as tracers. We can use them to fight cancer. There's all kinds of different things we can do with these things. Okay, so we're going to be looking at how you look at the periodic table. So the four most common and the four most important elements in biology are honk, right? How many people heard of this?
I have.
Honk, right? So honk really stands for hydrogen, oxygen, nitrogen, and carbon. And 96% of all the things that are biologically active, 96% of those things, I'm sorry, of all the things that are biologically active, 96% of those things, whether it's you or a flower or whatever, 96% of them are composed of those four elements, right? Hydrogen, oxygen, nitrogen, and carbon. These are the ones that we as scientists in biology want to talk about. The other things are important: calcium, phosphorus, potassium, sulfur, sodium, chlorine, and magnesium, a bunch of other trace elements, a bunch of other. They're all important because they do specific things for our cells and for our bodies and our tissues. But mostly what we want to look at are these four very common elements because they are greatly involved in everything that we do as a human, right? And really everything that we're interested in. Okay? So, can somebody tell me what's going on here? What's going on here?
The energy cycle?
Okay. And can you be more specific about that, Kelly? What do you mean? What do you mean by energy cycle?
I'm trying to remember the
They're metabolic pathways, right? There are two types of metabolic pathways. Can anybody name those two types of metabolic pathways?
citric acid cycle.
Nope, that's part
Um,
of the pathway. The two types of reactions that we see are called catabolic and anabolic. How many people have heard of those? Catabolic and anabolic, right? And so that's what's going on here, right? Catabolic is the breakdown of large molecules, right? And in this case, it would be whatever you eat today. Today's pizza is tomorrow's CO2, right? Because the carbons that are associated with the fats or the carbohydrates get broken down in the cycles to create and release energy so that the cells can do work, right? But the carbon is not needed. It's mostly the hydrogen that are needed, right? The carbon just bring those hydrogen to us. So the carbon have to get released, they have to get removed, and they get released through the process, right? And they become CO2. So just think about this, right? If we can think about what happens metabolically, it really is the intake of nutrients. The nutrients get broken up into simple molecules. Energy gets released. the cells do work. The cells go back and make up larger molecules so that the cells can grow and the body can grow, anabolic reactions, and energy gets used up, right? And this is what's happening, right? And we're going to talk about this in great detail, right? I'm going to expect you to know the end products and the beginning products of the entire cellular respiration process, right? It's one of the most important things, one of the important mechanisms you will learn. And I love talking about it. So we'll talk about that in unit 2, right? We're going to be talking about chemistry today, right? And so if we think about this, then you can see we take in food. Today's pizza is tomorrow's CO2. Are you with me? So just think about every time you exhale, you're releasing carbon that was once associated with your food. Isn't that cool? People don't think about that, but it's true. Good questions. So if we look at the periodic table, right? Um, if we think about the periodic table, right? Biology is only concerned with a few of these elements, right? They're over 117 and growing, right? Because with the super collider, different countries are making different elements. Those elements aren't lasting very long. They're not stable. They break apart very quickly. But they're creating all these new man-made elements, right? But we as biologists are only interested in a few of them. Now, for those of you who don't know how to read a periodic table, there are groups and there are periods in a periodic table, right? So groups, if you think about them, are the vertical alignment or the vertical arrangement of the periodic table. And those share common traits, electrons, right? And then periods, again, are the horizontal arrangement, right? And those share common traits or characteristics, right? And so it's important to think about how these things are arranged on the periodic table. Now, I'm probably not going to ask any questions about, what types of traits or characteristics do groups share and what uh, traits or characteristics do periods share, because I'm more interested in the entire structure of what we're trying to do and the entire picture. I'm more interested that you understand how these electrons and how these protons and neutrons are important in these atoms and in these molecules, right? So if we think about this, if we just look at these, you can see that these groups and periods are given names, right? Inert elements, sometimes known as the noble gases, the halogens. These are all the enes, chlorine, bromine, iodine. There's a bunch of them, right? Non-metals, there's other metals, there's transition metals, there's rare earth elements. There are alkali earth metals and there are alkali metals. And so if you think about these right now, a lot of interest is on the rare earth metals because they're the things that we need in order to make chips and other things that we are so dependent on. And really, when you think about this, China is buying up a lot of the reserves of these rare earth metals in the entire world. They're going into places like South America and buying these huge mineral rights, and then they're just taking all the rare earth metals. So really, what they're going to do is they're going to corner the market for rare earth metals, and that's going to put us in a position where you've never been in, right? Where we're just not going to have enough resources to. They want to keep selling to us, right? And if they corner the market, they're going to be able to charge us whatever the heck they want. And if we're going to want to buy a new phone, we're going to have to pay that price. Are you with me? So we have to be understanding of what's going on in the world, right? So if we think about these things, right, we can see how these different elements are arranged in the periodic table. I don't want to talk too much about this because I know most of you know this. So if you will review the slides that I didn't go over, and then if you have questions, you can contact me or you can ask or whatever in class, and we'll try to get them answered. But mostly I'm going to be testing you over kind of what's coming up next, right? and that is, what do these things, what are these individual elemental stamps on the periodic table mean, right? And so I know some of you know this, but why don't we review what these things mean, right? Real quickly, and then we'll call it a day because it's already pretty close to stopping. So if you look at carbon, C, and then you see a 6 and a 12, right? And so the 12 is the atomic weight, and 6 is the atomic number. What do those numbers really mean? What do those numbers really mean?
Atomic
Somebody tell me.
number is the number of protons, which is the same as the number of electrons.
Beautifully said, Kelly. What about the atomic weight? What does that mean? That's the mass. That's the mass of the atom, right? And so, if we were drawing, if we were drawing the nucleus of carbon, right? We could say, I'll draw it down here, that in the nucleus are 6 protons, right? They have a positive charge. And then also found in the nucleus are the neutrons. And how many neutrons do they have? 6. And the way you figure that out is you take the atomic mass and you take the atomic number and you subtract the atomic number from the atomic mass, and you get how many neutrons. Now, neutrons have a mass of slightly greater than 1. Protons have a mass of 1, right? And so therefore the atomic mass or the atomic weight is going to be 12. And then Kelly's already said that the number of electrons for the most part, right, have to equal the number of protons. So that's true. So if I was going to draw the outer shell or the outer shells of this particular structure, my first shell would have how many electrons? 2. So I'm going to put one here and one here. Now they could be anywhere because those are just holders for them. They're not there. They're actually orbiting around the nucleus, right? So the rule is biologically in that first shell, only 2 electrons are found, okay? But every shell or every orbital after that can hold how many? Up to 8. So how many electrons are we dealing with in carbon? How many total electrons? 6. So if I put 2 in that first shell, how many have to be in that second shell? 4. So 1, 2, 3. Sometimes I like to be kind of symmetrical, sometimes not. So there it is. That is really the electron model for carbon. But in order for that particular atom to be stable, how many electrons must it have in its outer shell? 4 more, right? So how many bonds then can carbon make?
4.
4, right? And that's what we're going to be continuing in our discussion on Wednesday, okay? So we will finish talking about atoms. We'll look at isotopes. Then we'll go into bonds, and we'll take care of that Wednesday. Are there any questions about